if I can do this multitask here. All right. It's a pleasure to be here, and what I want to do is just kind of give a brief introduction to some basic approaches to work with time-resolved connectivity, and then I'll go through a few examples where we've applied it to study brain disease. And then towards the end, I'll talk about some challenges, limitations, and possible solutions. In a sense, the most basic approach is to use a sliding window approach. What we often do is we use independent component analysis to decompose the data, and then we take those ICA time courses and apply, instead of looking at correlation between them the whole time course, we use a sliding window to get time series correlation matrices. You can see an example on the left there of how those look. And then what you do is cluster these into states, and that gives you some information about essentially stable connectivity patterns that individuals move into or closer to and further away from during the course of the 5 or 10 minute, et cetera, study. And you can also go further and estimate which state they're in. Here, for example, one individual started in state 3, then went to state 2, then went to state 4. Instead of using, assuming that an individual's in one state at any point in time, you can also treat them as sort of overlapping and continuous. And so here we have two states in sort of a state space framework. And so here's one individual starting here, and this is strongly in state 1 and strongly negatively in state 1, and the same goes for state 2. And they're moving through that state space sort of at different times. And we can quantify that in a vector here. So we have 2 points, one for each state, and it tells you the degree to which they're in that state at any point in time. And so if there's 3 states, then there's 3 numbers in that vector. And then we can calculate statistics on that vector to look for differences in patients versus controls, for example. When we looked at this in schizophrenia, one of the things that was encouraging is, well, first of all, we noticed that across the board we got negative values, meaning that we saw reduced dynamism, reduced dynamic range in the schizophrenia patients. The other thing that was interesting is this is sort of a global measure, right? So it's basically taking all of the states and then computing a summary measure from that. And you'll notice that regardless of how we decompose the states, whether we use k-means or PCA or spatial ICA or temporal ICA, all of those numbers were in the same direction. So it gives you sort of a nice global summary that captures the temporal information, and it isn't necessarily dependent on how you decompose the states. Each one of these approaches gives you very different looking patterns of states because they're using different assumptions. And that can be useful as well, but in this case it was somewhat invariant to it. And we also found that this information was also correlated with the negative symptoms in the schizophrenia patients. And one other interesting metric up here in the top left is you can calculate something called a hub state, which is like a sticky state. So basically, if someone gets into that state, they tend to stay longer, almost getting stuck there. You can think of it that way. Patients showed more hub states, and their time in those hub states was correlated with the negative symptoms. Here, we've looked across a lot of different studies now and found almost across the board that dynamic connectivity is more predictive if you're using a cross-validated classification framework than is static connectivity. Here we have a 3-way classification, bipolar, schizophrenia, healthy control, and we're comparing a static analysis to a dynamic analysis. And the classification for static is 60%, which is not bad because chance would be 33% here. But the dynamic analysis was 84%, and combining the two was close to 90%. Going a little bit further, we've looked at whether we can use accuracy, classification accuracy, to determine how many states we should estimate. Here you can see that, you know, going from 1 to 2 or more is across the board, you know, improving accuracy, with a slight improvement around, in this case, for a schizophrenia versus healthy control comparison, around state 4. Another thing that we asked was whether individual states were more or less informative about disease. And so if we use, again, kind of like a feature selection approach, here I've got a static analysis. Here I'm using all of the states for 3 or 4 or 5, so that's like this comparison here. But then these points up there are where we basically did feature selection and identified a smaller number of states that were predictive. And then we looked in unseen data and evaluated the accuracy. And you can see that the accuracy goes up when you do that. So you're basically taking the most selective connectivity patterns and using those to classify going forward. So let me just run through, just real quick, a few different examples. This was the first analysis we did with schizophrenia, where you see the five states here for patients and controls. Here's the difference. You can see that we get some interesting patterns when we look at just cell-wise differences. For example, state 2 and state 3 have a subcortical auditory sort of antagonism going on. And then if we compute for each individual state metrics, for example, the dwell time, the average amount of time they spend in a state when they enter it, we see that the healthy controls spend significantly more time in these modular states 1 and 2, patients in state 4 and 5. Okay, and I'm not getting any. Someone change the slide for me, please. Thank you. And we've looked also in sort of clinical high-risk patients as well, and we see similar patterns. So here's sort of an early, sort of almost the first episode group, and you can see, again, they're showing this reduced dynamism across the board, and then in these clinical high-risk individuals, so they don't have schizophrenia yet, but they are at risk for getting schizophrenia, you can see there's a trend in that direction, in particular in total distance and metastate change, reduced dynamism. Next slide, please. Here we have another example looking at a developmental study from the Generation R project. And so we have 800 children from ages 6 to 10. We have 4 states here. The younger kids tend to occupy more of this sort of less modular state, and then as they get older, transition to this more modularized state. But if we look in particular at autistic tendency, we see that those children tend to persist in this disconnected state, regardless of age. Next slide. Here's an example from ADHD, attention deficit hyperactivity disorder. This example, I think, provides some interesting observations that, depending on how we look at the data, we might get different conclusions. So if you look, for example, at the global measures that I mentioned before, we see evidence of increased dynamism in ADHD. But if you look within domains, for example, if you look within the default mode network, we see decreased dynamism. And so reconciling that, it's really a matter of sort of looking at different scales. And so I think there's a lot going on here and a lot we have to do yet to really understand what this is telling us. But the different scales, again, are giving you different windows into the data. Next slide. And one more slide, please. So if we transition to some examples from neurological disorders, one of the characterizations that's of interest these days is looking at vascular versus cognitive. Here we got Alzheimer's and subcortical ischemic vascular dementia. And can we differentiate those groups using resting fMRI? And if we look at it with this dynamic connectivity metric, we see that indeed we find that the SIVD individuals are showing a higher dwell time in this sort of less modularized state 3 compared to the Alzheimer's and the healthy controls. And then if we look over here at this state 4, which has this strong antagonism, positive and negative, we see that that state, if we regress out the disease categories, we see a strong relationship to cognitive scores, in particular attention and processing speed. Okay, can you jump ahead a couple slides, please? I want to go to, let's next slide. Just in the interest of time, I'm going to skip a couple slides. Next slide, please. So here, we're starting to try to synthesize across all of these results and really understand whether it's giving us specific information or general information. And you'll notice that sort of across the board here, we're seeing a lot of decreases regardless of which illness we're looking at. But the patterns tend to be, in terms of their magnitude, in terms of the directionality, there are, in some cases, we have stronger effects and weaker effects. In some cases, we have strong effects across the board. We're seeing some interesting patterns here. We still have a lot of work to do to try to get more at this and sort of characterize which of these can be useful long-term as markers of illness. Next slide. Next slide. So in the next few slides, I just want to touch on some ongoing work. This is a replication study we asked a couple years ago, basically taking 7,000 individuals and splitting them into 28 independent subsets and running a pipeline to see if we see canonical states and do we see things replicating. And what we found was that indeed we did see strong similarity across these different analyses, correlating at about 0.8. And likewise, the state that they tended to spend the most time in was consistent across the different analyses. You can see here it was in this state 3. Next slide. Motion is of interest, obviously, in anything that we're doing in brain imaging. Here, what we've done is looked at the results after we regress out motion. And we're finding that these explicitly dynamic states are relatively robust to motion. That's not to say they're not impacted at all, but you can see the distribution here across these, again, 28 different sets for those 4 measures. Next slide. One question that comes up quite often is, need a new battery maybe, is the window, the choice of the window size. And you know, how do we pick that, right? And there's been a number of papers that have discussed that in various contexts. One thing that we've been looking at recently is really trying to think of the window as essentially a filter on this dynamic correlation. And treat it as essentially a filter bank. So instead of looking at one filter, we look at a set of filters that cover essentially all the different window sizes from small to large. And then we follow with our clustering approach, as I mentioned earlier. And so then what we get is we get states out, and we also get their weights associated with each of the filter banks. So this is low frequency and high frequency. And so you'll see some here that tend to have a high value on the left side, which means they tend to be more static, less dynamic. And then the ones on the right, the one here that has high value on the right, those tend to be more transient or dynamic. So instead of using a fixed window that can sort of either smooth out the fast things or you know, or miss some of the slower information, we're trying to cover all of that if possible. Next slide. Another interesting direction is looking at these relationships between activity in a single time course versus correlation between pairs of time courses. And so one way to look at that is to compare, for example, a windowed ALF comparison in a single time course to dynamic connectivity matrices shown here. And what we're finding is that they don't always converge. Sometimes they converge and sometimes they diverge. So like with most of resting fMRI, the story is a bit complex, but I think there's some interesting information in there, and I think we should be looking at this. We also find that a relationship with cognitive scores in the length between, for example, dynamic ALF and dynamic FNC. Next slide. Okay, excellent. Okay, thank you. Another direction that I think we need to be heading in is to look not just at the temporal dynamic information. So here we have 3 different scenarios. We have fixed regions, and we have changes over time in the temporal link between them, versus having regions that move and no change, and they're linked in a consistent way across time, essentially static, versus changes in both spatial and temporal links. And so this, I think, is an interesting and important distinction. We've looked at this, for example, in a recent paper, and have found that we get, when we look at, for example, patient versus control differences in schizophrenia, we find quite a bit more sensitivity and quite a few differences in the spatial variability of these in schizophrenia. Up here on the top left, you can see we have a variability map, and you can see the static information in pink, which is showing a higher value in healthy controls versus patients, and the variable regions here in cyan, which show higher values in schizophrenia patients. Down here on the left, we can sort of color code regions that are white are sort of present in multiple states all the time. Ones that have a unique color tend to be unique to a given state over time. Okay, and then the last slide I have is really just to I tend to do things in a data-driven framework, just because we know so little about what's going on in this complex data. But we're trying to push towards, ultimately we want models, we want mechanisms, we want to understand what's going on. Here's an example where we've applied, again, a dynamic approach, but instead of just the time course pairs, we've used the temporal derivative as well. So we have at the top the state associated with the time course, and then below it is the temporal derivative. And so that temporal derivative is giving us information about whether we're learning, we're getting information flowing into or out of a given state, whether it's negative or positive. And so then this can allow us to look at models in the context of, you can think about it sort of an attractor, where you've got a state and another state, and you've got information flowing into or out of or between them. And we looked at this recently in a traumatic brain injury study, and we found some really interesting results where we see this sort of state A, which has this sort of pairwise but opposite patterned clusters. And so here's state A and state B in a healthy brain. If we look at 2 weeks after injury, we see that those are sort of decoupled from one another. 4 weeks after, we start to get partial recovery, and then 3 to 4 months after we have full recovery. So it gives us, I think, some really interesting ways to start to understand where the brain is disordered and looking at recovery. So where do we go from here? I think we still have a ways to go in terms of methodological improvements, looking at the impact of parameters and looking at the impact of confounds. There are a ton of validation studies, integration studies ongoing, which I think is heading in the right direction. I think for these time-resolved studies, we need more large-scale studies and look at synthesis across disorders. We need to start to build dynamic atlases, so not just looking at fixed atlases over time. Since we're dealing with brain function, things are changing over time in terms of how they're coupled. We also need to look at whether these are specific to disorders or unique to one disorder, especially if we're interested in this as a marker of illness. And then finally, I think we should be comparing whether these are informative about both dimensional as well as categorical changes in disease. So with that, I will close. Just to mention, we've released the approaches that I showed in the slide within a dynamic connectivity toolbox. It's part of our GIF software, which is available for download. Thanks to my lab and for doing most of the work here, and thanks to you for your patience in listening to me speak. Thank you very much.